This video provides instructions on how to thaw and plate BioIVT's cryoplatable hepatocytes. First, review the instructions for use guide for the hepatocytes. Then, gather the necessary materials and equipment. Instructions for use guides can be found on BioIVT.com in the Resources tab for each hepatocyte product. Place in vitro grow KHB and HT medium into a 37 degrees Celsius water bath for at least 30 minutes. Using a serological pipette, take 48 milliliters of the in vitro grow HT medium and add it to a sterile 50 milliliter conical tube. Take a vial of cryosuspension hepatocytes from the dewar and gently loosen the cap to release the pressure. Then retighten the cap. Place the vial in the cryovial rack set in a 37 degrees Celsius water bath. Maintain the vial in the water bath for approximately 90 seconds to thaw the hepatocytes. The vial should be swirled gently in the water bath and inverted several times. Once the frozen cell pellet releases from the vial wall, the cells are ready. Pour the vial of cells into the 50 milliliter conical tube of pre-warmed in vitro grow HT medium. Pipette 1 milliliter of medium from the conical tube to wash the cryovial for excess cells. Then pour it back into the conical tube. Gently invert the vial 3 times to mix the cells evenly. Centrifuge the cell suspension at 50 times G in a room temperature centrifuge for 5 minutes. Discard the supernatant by either pouring in one motion or aspirating with the vacuum pump. Loosen the cell pellet by gently swirling the centrifuge tube. Add 2 milliliters of in vitro grow KHB. Gently swirl the conical tube to resuspend the hepatocytes. The cells are now ready to use for the trypan blue method of cell counting. To learn more about the trypan blue exclusion method for cell counting, view the cell counting video located in the Protocols and Tutorial section under Resources at BioIVT.com. After cell counting is complete, determine the total cell concentration. The first step in determining cell concentration is to divide the total number of viable cells by the total number of squares counted. In this example, there were 8 squares counted. Viable cells per square is then multiplied by 10,000. 10,000 represents the hemocytometer conversion factor for the volume in the chamber. To account for cell dilution, multiply the product of the previous step by 10. The result of these calculations is the total number of viable cells per milliliter. In this example, the remaining volume of cell suspension is 1.9 milliliters because 0.1 milliliter was used for counting of the original 2 milliliter volume. To determine the total cell yield, the cell concentration of 3 times 10 to the 6th cells per milliliter is multiplied by 1.9 milliliters for a total cell yield of 5.7 times 10 to the 6th cells. The final concentration of the cell suspension depends on the research protocol. In this example, the target cell concentration is 2 times 10 to the 6th cells per milliliter. The remaining 2 milliliters of original cell suspension must be brought up to the final resuspension volume in order to achieve this target cell concentration. Determine the final volume required by dividing the total number of live cells by the target cell concentration. In this example, there are 5.7 times 10 to the 6th live cells. This number is divided by the target cell concentration of 2 times 10 to the 6th cells per milliliter. The resuspension volume is 2.85 milliliters. The original suspension volume of 1.9 milliliters is subtracted from the final resuspension volume of 2.85 milliliters in order to determine how much in vitro grow KHB must be added to bring the total volume up to 2.85 milliliters. Add 0.95 milliliters of in vitro grow KHB to the original suspension volume in the conical tube. The final cell concentration will now be 2 times 10 to the 6th cells per milliliter. The reconstituted cells are now ready to be used in a metabolism suspension assay. Now aliquot the cells in the plate being used. The target and reconstituted numbers may vary if a different plate type is used or for different study types. If you have questions, please contact BioIVT Customer Service.